Yeah, well, come back to our class, students. I hope you all are doing good. Okay, you learned it passive diffusion process. Mm, we just recollect what we discussed in last class, right? Okay. Then what we discussed the mechanisms of the drug absorption through the GIT. So first, let us recollect what are their main mechanisms. Three types are there, right? First one is intracellular mechanism, then intercellular mechanisms. Then what are those? Intercellular, intracellular, and vesicular type of transport. So where we are discussing about transcellular or also called as intracellular mechanism. In that we have four types. What are those? Passive diffusion, pore transport, ion pair transport, facilitated or mediated diffusion processes. In that we already discussed about passive diffusion. Next, let me discuss about pore transport mechanism. So what is there? Here your peritoneum. Then here this is the GI lumen. Then here this is the blood or systemic circulation, right? When you administer the drug through this pore transport, here some breakaging of your GI lumen will take place. From that it will enters or exits to this peritoneum to this blood. See here the arrow marks are towards peritoneum. From the blood through the transcellular pore, your drug will be entering to the peritoneum. See here, this is the large pore and this is the small pore. There is a chance to form a pores in between the GI lumen for entering or exiting of your drug from peritoneum to the blood. That's why the name came it as pore transport. The name itself indicating that in the mechanism of transportation of the drug, the GI lumen pores will be formed in between the GI lumen. Why? Because the entering or exiting of your drug molecule from the bulk to the site of administration. Okay, then what it indicates, also known as convective transport or bulk flow or filtration. See here, your particles will be bulk. Bulk means more than one. It contains number of particles. That is nothing but your bulk. Your distributed, your dispersion medium or your aqueous medium is having more than one particle. So bulk of the solution will be formed. So this pore transport is also known as convective transport or bulk flow or also called as filtration. Remember here, important in the absorption of flow, low molecular weight, less than 100. Low molecular size, small than the diameter of the pore, and generally water soluble drugs through narrow, aqueous filled channels or pores in the membrane structure. So what are the examples? Urea, water and sugar. So coming to this, how the drug will be and which type of drugs are morely transported through this pore transport mechanism. So which type? The drugs which are showing low molecular weight, that is how much it is, less than 100. The molecular weight, you have units of Dalton. So here there is not mentioning any Daltons, like so less than 100 Daltons. So then only your drug will choose this type of pore transport for achieving good GI absorption, right? So here the examples are urea, water, sugar. The water solubility is more for your drug, then only it is achieving this type of pore transport. Urea is also is a water soluble moiety. So that's why these three are the examples for pore transport. Remember this? Okay. The next one is the driving force for the passage of drugs is the hydrostatic or the osmotic pressure difference across the membrane. Hydrostatic pressure will be developed across the GI lumen. For passage of drugs, it should develop some gradient. It should develop some driving force. That should be the hydrostatic driving force or pressure difference across the membrane. That is about pore transport, the mechanism of pore transport. And next, ion pair transport. So the absorption of drugs, like the name itself indicating, what is that? Your drug is having some ions, positive ions or negative ions. So how the drug absorption will take place, we will discuss it. Absorption of drugs like quaternary ammonium compounds having positive charge and sulfonic acids having negative charge, which ionize. Ionization means what? Converts into the molecules under all pH conditions is known as ion pair transport. Once again you come back. Absorption of drugs like quaternary ammonium compounds positive charge and sulfonic acids negative charge, which will ionized or which will converts into molecules under all pH conditions with irrespective of pH. That means in stomach you are having the pH of 1 to 3, in intestine you are having the pH of 5 to 7. So these positive and negative charged ions will be ionized with irrespective of pH conditions. That means it should be ionized at all pH conditions, is known as ion pair transport. See clearly, ma? I am speaking, I am telling, explaining very clearly. You all should know the mechanism. Such neutral complexes have both the required lipophilicity as well as aqueous solubility for passive diffusion. Such phenomenon is called as ion pair transport. Then what do you mean by hydrophilicity, lipophilicity, hydrophobicity, right? Hydrophilicity means water loving nature. Hydrophobicity, so these may be solubilized in organic solvents. Hydrophobicity or also called as lipophilicity. Such phenomenon is called as ion pair transport. Propranolol or LOL's antihypertensive drug is a basic drug that forms an ion pair with oleic acid, is well absorbed by this mechanism. See what is the example? Propranolol. It is an anti-HT hypertensive drug. It is a beta blocker. It acts by blocking beta. Okay, it acts by blocking beta receptors. You have alpha beta receptors, no, in case of hypertension. So it acts by blocking of the beta receptors. Okay, is a basic drug. It forms an ion pair with oleic acid, is well absorbed by this ion pair transport. SEMA here, cationic drug and endogenous anion. I'm always telling you that site of administration to the systemic circulation. The unchanged movement, we have to note it down under absorption, right? So the entry of your drug from GI lumen to the blood. See, cationic drug, endogenous anion. See, it forms a neutral ion pair complex. Neutral means it's a constant. See here, positive charge and negative charge will combine here and it forms a neutral ion pair complex, and this complex will be passed to this blood or systemic circulation via the membrane by the mechanism of passive diffusion. Okay, the dissociation of complex will take place in blood. See how clear the mechanism will be. See ma, you all should observe the picture very clearly. You just stick on to your brains. Cationic drug having positive charge, endogenous anion is having negative charge. These two are combined together and forms a neutral ion pair complex. See clearly under GI lumen. So these complex will be passaged through passive diffusion across the GI lumen. it enters to the blood or systemic circulation. Then it will get dissociated. Your complex will get dissociated to positive and negative and free drug, which is unbound drug. Okay, this is about your ion pair transport. Clearly explained it, right? And next one is facilitated diffusion. We discussed about passive diffusion, ion pair diffusion, pore transport, and fourth one is facilitated diffusion. These all are comes under intracellular mechanism or transcellular method of mechanism for GI absorption. Facilitated diffusion, it is a carrier mediated transport system that operates down the concentration gradient, downhill transport, but at a much faster rate than can be accounted by simple passive diffusion. So what do you mean by diffusion? The drug will be transported from high concentration to the low concentration. That is nothing but uphill transport. That is nothing but uphill transport, which is described by Fick's very clearly. Fick's first law of diffusion. So here in case of facilitated diffusion, it is a carrier mediated transport system that operates down the concentration gradient. That means your drug will transported through from low concentration to high concentration, and it produces some concentration gradient around the GI lumen or membrane. Lumen is also called as membrane, so don't get confused, right? This is called downhill transport but at a much faster rate than can be accounted by simple passive diffusion. You may attain much faster rate of diffusion as compared with normal diffusion. The driving force is the concentration gradient, hence a passive force, so no energy expenditure is involved. The process is not inhibited by metabolic poisons that interfere with energy production. So they explained with the example of vitamin B12. What do you mean by vitamin B12? It is a cyanocobalamin vitamin, right? So the drug will develop some driving force around the GI lumen. So how the mechanism will be? This is the GI lumen, this is membrane, and this is what? Blood. Are you noting? Are you noticing everything? So just note down, ma. Gastrointestinal lumen membrane. This is the blood. So see here the structure of vitamin B. This is an intrinsic factor. This is a carrier. Okay. So the B12 intrinsic factor IF carrier complex will be formed here. I may discuss about intrinsic factor and all in the next classes, right? Clearly. So first you just remember as intrinsic factor IF. Vitamin B, intrinsic factor, and as a carrier. These three may form a complex here. See ma, BIF carrier complex. Okay. When this complex will be transported through this membrane, your carrier may get attached to this membrane, and the dissociation of intrinsic factor and free vitamin B12 will take place in the blood. Okay, here vitamin B12, intrinsic factor and carrier are joined by complex, and when it will get passed on through this membrane to the blood, the carrier will get attached to this membrane or barrier. Then when it enters into the blood, the free vitamin B12 is dissociated as complex. See, dissociation of complex. This is the facilitated diffusion of vitamin B12. And the comparison of rate of absorption, how the rate of absorption versus drug concentration plots for passive and carrier mediated transports. Okay, this is the rate of drug absorption and this is the concentration, how much, 10 micrograms or 20 micrograms or 30, so on, 100. Okay, this is the concentration of drug at the absorption site. How much is the drug concentration available for its proper absorption and rate of drug absorption, right? Here you may observe one straight curve and one curved curve. This straight curve possesses passive diffusion. See ma, here it follows zero order mechanism. I will tell you what is zero order and first order. Okay. Next, then this is the carrier mediated transport. It is showing some downwards to this passive diffusion. So your carrier mediated graph is like this and your passive diffusion graph is like this. So this is the comparison of rate of absorption of passive diffusion as well as carrier mediated transport of diffusion, right? This is about your passive diffusion processes. And then next, active transport processes. These transport processes, active means definitely they should be active. So they need some energy, simple. So just remember it as simple thing, active. So if they should possess some activeness, they need some energy to speak something. We need to take some food and all, then only our voice will be loud. Same like that, it needs some energy, that is in the form of ATP. What do you call by ATP? Adenosine triphosphate. To move the drug molecules from extracellular fluid to intracellular fluid. There are of two types. You already know that. Primary transport and secondary transport. Active transport is of two types. Primary active transport and secondary active transport. And then secondary active transport is further divided into two types, ma. Symport, antiport. Symport, same. See here, S stands for S. Symport, that is same. That is nothing but co-transport. There is a coordination. There is a symport, that is the same. So I am telling you more than one word, more than one. Those are called synonyms. You just remember it, ma. For remembrance only, I am telling you for easy remembrance. Symport, that is co-transport. And next, antiport, counter-transport, counter, opposite. Okay. Symport and antiport. Adenosine triphosphate is a complex organic material or chemical that provides energy to drive many processes in the living cells. For example, muscle contraction, nerve impulse propagation, and chemical synthesis, right? Adenosine triphosphate is a complex organic chemical that provides energy to drive many processes in living cells. Example, muscle contraction, nerve impulse propagation, and chemical synthesis. These processes, they need some ATP to process themselves. In that, two types now, primary active transport as well as secondary active transport. First, we will discuss about primary active transport. Come to this. Primary active transport. In this process, there is a direct ATP requirement. Primary means fast. Primary active transport. In this process, there is a direct ATP requirement. Moreover, this process transfers only one ion or molecule in only one direction. This is the very, very important thing here. The process transfers the ions only in one direction, not for any other direction, and hence it is called as uniporter. Uni means one. Example is absorption of glucose. See ma here, primary active transport, and this is secondary. This is, we just leave it for some time. So now we are telling about, I am telling about primary active transport, ma. Here it requires energy. See here, in this process there is a direct ATP requirement. Okay. Moreover, the process transfers only one ion or one molecule in one direction. Okay. And hence called as uniporter. See, only one direction it is transporting its molecules or ions, Na plus ions. See here, ATP requirement is there and it is transporting in one direction only. So this is called active transport. Okay, this is called active transport. Then what is this image is showing? I already told you, antiport and symport. Anti, co-transport, symport. Symport means same, as for same. So same directions the molecules are traveling. See here, antipode, counter, see opposite charged, opposite directions, see opposite directions. Antipode, the molecules are distributing through opposite directions. Sympode, the molecules are distributing in same directions. Co-transport, counter-transport. And come to the secondary active transport. In the secondary active transport, there is no direct requirement of ATP. That is, it takes advantage of previously existing concentration gradient. The energy required is transporting an ions aids transport of another ion or molecules either in the same direction or in the opposite direction accordingly. This process is further divided into. Okay, then this is next step. First, we would like to know what is secondary active transport. Primary means they have the capability of moving that ions and all in only one direction. But in case of secondary, see here, no direct ATP requirement is there, and this is having the ability to transfer transporting ionates to another ions or co-transport or coupled transport either in same directions or whatever the direction, same or opposite directions, but it is having the capability of promoting more than one directions of ions. In case of primary, they have ability to transport of ions in only one direction. So next one is symport and antiport. These two categories are comes under secondary active transport. Symport involves the movement of both molecules in the same direction. Sodium plus glucose importer. See here, in the same direction. Symport involves movement of both molecules in the same directions. This is symport SEMA. This is sodium molecule and this is glucose. These two are in same directions. This dark pink long line and this light pink long short line. Okay. Next, antipode, counter transport involves movement of molecules in the opposite directions. Examples are H plus ions using the Na plus gradient in the kidneys. See ma, this is K plus ions, it is going down, and in case of sodium ions, it is going up. So these are showing like opposite directions. Ion transport will be, ions will be changing into opposite directions, right? So this is about primary and secondary active transports. Now then, types of active transport is combinedly explained in this graph. See ma here. See very clearly it is concentration gradient. It is developed around the GA lumen. Everyone is clear this? Next one is, this is primary active transport and secondary active transport. In these, ions can be transported in only one direction and it requires some ATP. These two are main, and it doesn't require direct ATP for transporting of ions in case of secondary. And in symport SEMA, these two ions will be traveling in same direction. See, this go down and this arrow is also down. So this is symport. And in case of antiport, see, this arrow is down and this arrow is up. So the ions transfer is in opposite in case of antiport. So secondary active transport does not require direct ATP for transportation of ions. In case of primary active transport, it directly requires ATP to transferring ions. Okay. So what is the main comparison of between active and passive transport? See, this is passive, that is downhill. This is active, that is uphill. So in case of downhill transport, this is GI lumen, GI barrier, blood. Your drug particles will be high drug concentration to the low drug concentration. So the drug particles are moving from high concentration to the low concentration, high to low, that is downhill transport in case of passive diffusion. So then in case of active transport, that is uphill transport, the drug molecules will be transferred from low drug concentration to the high drug concentration. See, ma, once again I will tell. In case of passive diffusion, it is also called as downhill transport. The drug molecules will be transferred from high drug concentration to the low drug concentration. In case of active transport, it is also called as uphill transport. The movement of drug particles from low drug concentration to the high drug concentration. This is the major difference between active and passive transport. Okay, till now what we discussed about intracellular transport, or also called as transcellular transport. In that, again, passive transport. Once again, I am showing the slides. In that, see here, transcellular or intracellular, we discussed till now. In that, you have transcellular and intracellular transport is categorized into two types, active process and passive process. Under passive diffusion, you have four: passive diffusion, pore transport, carrier mediated transport, and facilitated diffusion. Under active transport, you have primary active transport, secondary active transport. Under secondary active transport, again classified into two types, symport, antiport. We discussed till now these many transport mechanism. This is all about transcellular or intracellular transport. Please don't get confused. Next class, we will discuss clearly about intercellular or paracellular transport. Okay. Okay then. Next class we will meet.